Thank you very much for the invitation to give this presentation on considerations related to estrus synchronization in sheep. There have been a number of recent reviews that provides a historical overview about progestogen systems that over time, the last 70 years, there also was another review recently that looked at an update of estrus synchronization in sheep in 2017, another one on the use of prostaglandins to control estrus cycle of tissue sheep, and then one in 2012 on hormonal control of reproduction. I did prepare an overview of estrus synchronization in 2000 at the request of the American Society of Animal Science, and this review here will be an attempt to update that information that was presented there. So, the historical background of estrus synchronization. Efforts to control estrus synchronization, to control estrus ovulation in sheep date back to the work of Dutt and Catheter in 1948. They used daily injections of progesterone for 14 days to control the estrus cycle, and they were able then to successfully make use 2 days after termination of the last treatment. This was further refined by Dutt and Robbins in 1952 that showed injections with PMSG at the end of treatment induced estrus and ovulation during seasonal and estrus. With further refinement of those techniques and building on these principles, when highly active progesterone analog, fluorogesterone acetate and methoxyprogesterone acetate were developed, and these then were consequently led to the development of vaginal passivary sponges that were impregnated these progestogens in 1965. This then was followed by the development of progesterone-impregnated silicone elastomer devices that could be used to control hormonal release. An alternative approach to synchronization was developed with identification of prostaglandin estrus alpha as a luteinurgic hormone in sheep. So that was done by McCracken in 1972, and it was demonstrated that a single injection was successful in inducing luteolysis. It was further noted that the luteum is responsive between the days 5 and 14 of the estrus cycle to injection of prostaglandin. And again, there was a development of effective analogs of prostaglandin to use in those systems. Just a quick review of applications of estrus synchronization. Foremost and most basic can be used to synchronize lambing and help with in the production system that way. It can also be used to induce out-of-season breeding in support of accelerated mating systems. And then it will facilitate the use of artificial insemination, especially timed AI, and that again gets used as part of embryo transfer. So the traditional system we referred to earlier is a progesterone-based system, which involves a 14-day insertion period with an optional injection of PMSG at insert removal. And this tends to bring the sheep, the ewe in estrus 2 days after implant removal. Again, the system can be used for out-of-season breeding, introduction, and it's suited for the use of artificial insemination. This then gives you a quick overview of a progesterone-based system using laparoscopic AI. This was a comprehensive study from Argentina that used 2,300 sheep, some young ones as well as some large number of older ewes, and these trials were done during seasonal breeding, April and May, using MAP sponges that were inserted for 14 days, and followed by PMSG treatment as sponge withdrawal. There were 2 systems of AI used. One was laparoscopic AI, 12 hours after the ewe was detected in estrus, or timed AI, 16 hours after the end of treatment. There were really no practical difference in terms of pregnancy rate between detected estrus, which achieved 63% pregnancy rates, and timed AI, which achieved 59% pregnancy rates. Pregnancy rates tended to be lower in the younger animals. This study then gave an overview of comparing CEDAR or progesterone-based systems with progestogen-based system or MAP sponge, and you can see here that in this study the cervical AI and laparoscopic AI pregnancy rates were higher using cedar sponges compared to the MAP system, ranging from 60 to 65% pregnant to AI, laparoscopic AI, compared to 50% here. So in this study, there was a difference between the two systems. The other traditional system is a prostaglandin-based system. There's a need for use to be cycling for the system to be applied. Traditionally, there have been injections of 9 to 14 days apart to catch the use in the right stages of the estrus cycle to respond to prostaglandin. Generally, they're considered to provide less synchrony than progesterone-based systems. However, there are new protocols now that use shorter intervals. The 7-day, or called synchromine system, that has also been used for laparoscopic inseminations. So these are then some conceptions made following estrosynchronization with either two types of prostaglandin. You can see this is prostaglandin after alpha here at 50 milligrams, and the light blue column, which is prostaglandin analog at 100 micrograms. And you can, study was done with Greek dairy use, and you can see under natural conditions, they achieve pregnancy rates of about 63%. And you can see that synchronization with both prostaglandin analog here and the MAP system achieved similar pregnancy rates here. The study was expanded to look at the use of artificial insemination as well, and you can see that the prostaglandin analog system performed considerably better than the MAP system for the artificial insemination protocols that were implemented. This is yet another study that paints a little bit different picture comparing progestogen systems to prostaglandin systems. In the blue here, you have your prostaglandin systems again, using 2 times injection of prostaglandin 11 days apart, either with a 5 milligram dose of prostaglandin or a 10 milligram dose of prostaglandin. This was compared to a MAP system where sponges were inserted for 8 days, followed by prostaglandin at the end of the insertion period. And you can see that in this case, the sponge system looked considerably better. It achieved higher pregnancy rates than the prostaglandin system. This then shows the pregnancy rate and prolificacy after a short duration prostaglandin protocol, the synchroven system we talked about that used a 7-day injection interval. And they were looking both at AI to observed estrus and compared it to a timed AI that was done 42 hours after the last injection. And you can see similar pregnancy rates were achieved, making this a suitable system to apply for timed artificial insemination using prostaglandin. This is another study looking at prostaglandin and comparing different injection intervals ranging from 10 days to 16 days. And as you can see here, better conception rates were achieved with a longer insertion period, especially the 14-day period, which achieved conception rate of 55% and 52% for fertility. Apart from prostaglandin and progestin systems, there are also alternative systems, which is for once the norgestimate cattle synchronization system, which uses an ear implant. And the system has also been adapted for cattle. A similar protocol is being used as it is used for sponges and cedars, and is probably used more in countries that did not have the proper sheep synchronization system in place. The other option is melanogastrel acetate, which is an oral formulation of progesterone and has been used and developed to use to suppress estrus in feedlot heifers, commonly used in the United States. This product is mostly used in when no other sources of commercially available progestogens are available, and it has been used in the application of lambing management and out-of-season breeding of ewes. This is then a slide that shows the use of norgestimate. and comparing it to a cedar insertion, and you can see both systems achieved similar levels of out-of-season estrus in induction. However, you noticed here that the ovulation rate, that the pregnancy rates were quite a bit lower than the estrus synchronization rates here. This then is another study that compared MAP sponges to norgestimate sponges. You can see comparing dose rates of 1.5 milligrams norgestimate and 3 milligrams of norgestimate. The observation here was that the percent of use coming estrus tended to be higher under the norgestimate system than the MAP system. But time to estrus was comparable, and pregnancy rates were not significantly different between treatments, partially based on the relatively small number of animals used in the study. And this then gives an overview of the use of MGA for out-of-season estrus induction and synchronization. There was a comprehensive study done in the US in 1996 that used about 1,650 ewes in 10 different trials during seasonal estrus, May and June. The observation there was that an 8-day feeding period was more favorable than an 11 to 14-day period, and it was observed that co-treatment with Zeranol was most effective when it was provided 54 hours after the last MGA meal. They compared Zeranol and estradiol and noticed that both systems, both products worked equally well. This is another MGA feeding trial here dating back to 1994 that looked at MGA feeding for out-of-season estrus in induction. You can see in the normal, with untreated ewes, you get estrus induction. You get an incidence of estrus over 12% with 10% lambing rates. This was substantially increased using MGA feeding here in yellow and orange, and you can see estrus induction reached as far as 70% here, and lambing rates went from 36 to 50% using MGA-based system, either with Zeranol or PG600. There were a little bit higher inductions and lambing rates achieved using norgestimate, and you can see induction rates went as high as 90% and lambing rates as high as 60% under the system. This is a study we conducted at Virginia State University looking at short duration MGA feeding for 5 days and combining with prostaglandin F2 alpha injection. You can see that if MGA feeding, short duration feeding, is combined with prostaglandin injection, you get acceptable induction and pregnancy rates out of season. So the final alternative system I want to mention is a male or ram effect, which involves the strategic exposure of rams to N-estrus use. It's been a system that's been in place and used by farmers for quite some time. It's primarily used to advance the onset of seasonal breeding, but it now is being explored as a substitute for gonadotropin treatment in progesterone-based protocols. Here's an example of one of these comparisons. This is using the ram effect in conjunction with the progestin treatment. MAP insertions, study used 3 insertion periods, 6, 10, and 14 days, and compared it to a non-inserted group that had also been exposed to rams. And you can see that the ram effect in conjunction with the treatment of progestin achieved markedly higher use coming to estrus than just the ram effect by itself. It also tended to reduce the period of days to estrus, combining these two treatments. This study achieved about 60% out of season pregnancy rates in these animals. The next part of the talk, I would just like to talk about limitations and constraints that are associated with estrus synchronization. Firstly, there's the health concerns that are with the use that is related to vaginitis caused by vaginal inserts. This is caused by the physical presence and retention of fluids, and is at times compounded by the release of the hormones. And these findings tend to be similar for both sponges and cedars, even so there's anecdotal evidence that the secretions resulting from CDAs are not as purulent as those of sponges. So this results, that vaginitis comes from a buildup of bacterial bacteria that are present after a relatively short period of insertion, but tend to be cleared out pretty quickly after the inserts are removed. These infections, when they occur, respond favorable to antibiotic treatment, but currently there are some major concerns now with the increasing in antimicrobial resistance and their use in livestock production systems. Another limitation and constraint are the regulatory hurdles. The availability of devices is really dependent very much on region and country, and has been subject to change. As a good example, in the United States, there have never been sponges approved for use. Similarly, sponges are really no longer allowed for use in Australia as of 2016. In contrast, CDAs have only recently been approved in Europe. Another concern is availability of PMSG. There is concerns by animal welfare groups about the production of PMSG meeting animal welfare standards at these what they call blood farms, especially in South America, that don't seem to have a very high level of animal welfare. And so this has moved production out of Europe into outlying countries, and there's pressure on countries to no longer on companies to no longer use PMSG. So now there's a concern or interest in developing alternatives to PMSG, and kisspeptin, glycerol, and GnRH have been examined, and I will touch briefly on GnRH later. And then there are environmental concerns, since there's a public perception against the heterologous compounds used in livestock production. This would be hormone analogs, equine chorionic gonadotropin and human chorionic gonadotropin being used outside their own species. And then there is the issue of drug disposal, environmental pollution, especially the aqueous contamination from residual progesterones leaching into indigenous fauna. Steps to take or considered are to decrease the amount of product that is being used, or start looking at the reuse of product to deplete the amount of progesterone present at disposal, and also look at the development of new delivery systems. Finally, there's a biological shortcomings. It's been well documented that there are lower pregnancy rates to synchronized estrus than there are to natural estrus. And a variety of issues that are related to that. One of them is the reduced progesterone concentration that are present at the end of an insertion period. There are also issues with sperm transportation in prostaglandin synchronized cycles related to the smooth muscle contractions. And finally, it's well documented that the follicular dynamics are altered in an estrus synchronized cycle. So what approaches can we adopt then for techniques to reduce environmental concerns and the animal welfare issues? Firstly, we have to look at recycling devices. We can look at reducing the amount of product that's available in these devices. We might want to look at shorter treatment period, again, using the amount of product that is being put out, and then maybe we need to explore alternative compounds. I want to just touch on a couple of these. This picture here then speaks to progesterone concentrations that are being achieved by using single use, double use, and 3 times used CEDAR devices. You can see that after the first and second use, progesterone concentrations remain relatively high at the end of insertion period. But you can see here that in a 3 times used product, these levels drop quite a ways, and this is then reflected in a decreased or tendency to have lower pregnancy rates when a 3rd time used product is used. But apparently a second use product is a viable alternative and will help to reduce the contamination. The other point I want to touch briefly on is the use and exploration of GNAH now to take the place of PMSG. This study then looks at GNAH taking the place of ECG in a 5-day period. Here you have the control group with your standard 5-day protocol. The second group, the GNAH group 1, also has the 5-day insertion period followed by GNAH 56 days after seeded removal. And then you're going to have a second group where there was an injection of GNAH given at the end of, at the onset of seeded insertion, and then again 56 days later. The results were as follows: that we had a similar degree of estrus synchronization with all three systems. The onset of estrus tended to be a little bit later, about 3 days, 5 days later in the GNAH groups. However, the fertility was very similar in all the systems, which might help making GNAH a viable alternative to PMSG. There's another study then that looked at the use of GNAH in a synchro-woven protocol. There's your straight synchro-woven protocol here, and prostaglandin given at day 1 and 7 with timed insemination thereafter. And then this is in the GNAH system. GNAH is given at 24 or 36 hours after the second prostaglandin injection. And you can see here that the pregnancy rates tend to be a little bit lower, especially when only single injection of GNAH was used. So some more exploration is needed to refine the synchro-woven protocol in conjunction with GNAH. So some of the final thoughts to bring this review to an end is that there's a need to balance the maximum production outcomes with environmentally sustainable system that can be accepted by consumers and the society as a whole. At the same time, we'll have to educate consumers of the relevance of assisted reproductive technologies towards enhancing genetic diversity, increasing biosecurity and animal welfare, and improving efficiency of production. With this, I'm glad to entertain any questions you might have.